Okay, group. So this is Ed Puzzle 3 for our cell division notes. This one's going to go over those kind of stop signs that we talked about in class about how the cell knows to keep going through cell division. So we're going to talk about how the cell knows to stop, how it knows to pause, and then what happens when it does not take those hints from the cell and it continues to grow rapidly or divide rapidly. Okay, so the cell cycle is regulated by these regulatory proteins inside and outside of the cell. So, what these proteins do is they basically say, okay, these cells need to go ahead and go through cell division, or if everything's good and you have, you're just kind of maintaining life, then you can turn that cell division button off. So, you have these cyclins and they are these proteins, and their job is to say, okay, we need to go through cell division. We're too big. We're ready to go, or we're okay for now. These controls can be turned on and off. So when they are turned on, again, it's because there needs to be some healing involved. So in the event of like a broken bone, this indicates because cells will grow or they will start to go through cell division when they're not touching any other cells. So when there's a gap in between like your bone or in between your skin and those cells aren't touching each other anymore, then those proteins are able to detect that and they turn the control on that says, okay, let's go through cell division. They will stop or the cell division will no longer keep going once healing is completed and those cells have started touching each other again. So that's the indicator that everything is good and cell division does not need to keep going. So the discovery of cyclins are going to be those proteins that regulate the cell cycle. So again, they're the ones that say we can stop or we need to keep going with cell division. And how this was discovered is they were looking at a certain protein inside of fertilized clam eggs during the growth and development of a clam. And in doing so, they accidentally found another protein, which would then become these cyclins, what we describe as cyclins. And what they saw is that these cyclin levels change throughout the cell cycle. And so we will do an activity, the rise and fall of cyclins, in class together. But that's what we're looking at, is that the cyclin concentration will go up and then it'll dip after mitosis is done, and then it'll start to increase, and then it dips once mitosis is done, and it goes up and it dips after mitosis is done. So you see this rise and fall. You see this very nice pattern of these cyclins being very active or the concentration increasing during mitosis, and then once cytokinesis happens, it will drop off again, and then it'll slowly start to rise again. And so every time that this is happening, it's basically showing that during cell division or just prior to cell division, during interphase leading up to mitosis, the protein levels are getting higher, and that's telling the cells, okay, we need to go ahead and start the division process. Now, there are also these things within mitosis, the stop signs that we talked about in class, and these guys are called regulators. So the proteins before the cyclins, these are proteins that say, okay, let's get this whole cycle going. Let's get the whole cell cycle so that our cells can divide. Regulators, these are going to be proteins that are internal to the cell cycle that basically say, okay, are we okay to keep going or do we need to stop? So there are internal regulators. These are going to be the ones that they're inside the cell. And so they are looking at what's going on inside the cell. Are the conditions good inside? And they're going to allow the cell cycle to happen as long as the checklist has been marked off. Has the cell grown correctly? Has the DNA been done correctly? So these guys are internal, they're inside the cell. The external regulators, these are little proteins that are on the cell membrane. So we talked about how the cell membrane has proteins or cholesterols hanging off of them. They respond to stuff that's happening outside the cell. So what are the conditions like outside of the cell? They can speed up or slow down the cell cycle depending on the conditions. And growth factors are going to be the thing that stimulates the growth and division of cells. So growth factors would be like outside of the cell, things like pH, temperature, environmental conditions, things like that, where if the outside conditions are not perfect for cell division, then cell division will stop or slow down extremely. So growth factors are also present in embryonic development and in wound healing. So this is where like, okay, you have a cut in your skin and the external regulators will basically during a cut, if the cells are like this and they get cut apart, well, they're no longer touching each other. They're like grasping for each other. So how they get to come back together is they say, okay, let's go ahead and divide. And then they slowly grow more cells toward each other, closing up that wound. So these are the growth factors. They stimulate that growth outside of the cell. All right, the internal regulators, there are 4 of them that you need to know. You do need to add this to your notes packet, I believe. Let me triple check that. Ooh, wrong way. Yes. No. So you'll need to add these to your notes, but there is a notes area for it. So I will get rid of it. All right, so there are internal regulators. These are the stop signs inside of the cell where it says stop. Is everything okay to keep going? So the first one, checkpoint 1, is during the G1 phase. So it's not quite to the end of G1 between G1 and S. It's almost to the end of G1. And what the G1 checkpoint is looking for, so kind of its checklist, is any of the DNA that's going to go through DNA replication, is it damaged? Checkpoint 1 is also looking for, has the cell grown enough? Once checkpoint 1 has been passed and the cell can go into the S phase, toward the end of S phase, there is checkpoint 2. And during checkpoint 2, this is where this regulator is really important because it asks, okay, is the DNA replicating correctly? Are there any errors? Can we cut out those errors? If no, then we need to abort the mission. Checkpoint 2 allows for the cell, if all of these answers are yes, everything's good, DNA is good, there's no errors, the errors have been cut out, then it allows for the cell to continue into the 3rd checkpoint region, which is the G2 checkpoint. Once we get to the end of G2 and we're getting ready to go into mitosis, the checkpoint asks, is the DNA okay? Are there any damages? Do we have all of the chromosomes necessary? Are there enough cell components? So do we have enough cytoplasm? Do we have enough endoplasmic reticulum? Do we have enough ribosomes? Do we have enough lysosomes? And if everything checks out, then it'll allow the cell to continue on into M phase or mitosis. Checkpoint 4 happens inside of mitosis, and it happens during metaphase. So it's after metaphase, so the chromosomes have lined up in the middle, and the spindles are all attached to the chromosomes. Before it moves on to anaphase, there is a checkpoint here to make sure that the DNA has been connected to the spindles, because if any of those chromosomes or those sister chromatids that are attached to each other, if any of them miss the spindle has not connected to them, then that means instead of separating and each sister chromatid going to opposite sides, that means that one side is going to get the entire chromosome and the other side will get none. And that can lead to significant mutations down the line. So these checkpoints, again, they're just like little guys. Like that's how I picture them, as little emoji guys with like a stop sign or their hand up, and they have their little check clipboard, and they are asking the cell, or they are checking the cell, okay, is everything okay? Is it able to move on? If not, then we cannot go on with this process. So when the process keeps going, even after the checkpoint has been, so the checkpoint is like, is everything okay? And somehow it misses that everything is not okay. This is how we can get cancer. So cancer is uncontrolled cell division. And the reason that cancer is so detrimental to the human body, or whatever organism may have it, is because those uncontrollably dividing cells, they start taking over the areas where these other specialized cells should be. So for example, if somebody had like liver cancer, those cancerous cells are starting to take over the area where the liver cells should be, and now the liver cells can no longer function properly, and so the person with liver cancer would start to lose liver function. So what cancer is a disorder in which the bodies lose the ability to control the cell cycle. Why this happens is because cancer cells, they are not responding to those signals that say, okay, we don't need to divide right now. They are ignoring the regulators. And so as a result, they just keep dividing, even though the regulators are saying, stop, like, don't do that. They just keep going and they basically go rogue. A tumor is when these cancer cells keep replicating and you have a collection of them and it creates this mass of cells. So that's why typically a tumor is going to be your first indicator that you have cancerous cells, because again, one cancer cell isn't a huge deal. It's when you have a huge clump of them that's when you're going to start to feel uh, the detriments or the negative impacts of that. Now, not all tumors are bad. You can actually have a benign tumor. So these are non-cancerous tumors, and what that means is they will not spread to surrounding tissue. They are just very localized and they're not really hurting anything. If you have ever had a wart, a mole, or a freckle, you have technically a tumor. So it's just a collection of those particular types of cells that are all, a wart would be a collection of the keratinized skin cells, and then freckles and moles, those are going to be collections of melanocytes, which are the things that give your skin color. So a benign tumor is not a big deal. Sometimes it can be uncomfortable and you can get them removed if you are like self-conscious about them, or they are causing, like, if you had like a wart in your armpit or something and it's causing friction, you can get that removed. A malignant tumor, however, those are the cancerous ones. Those are the ones that will spread to the surrounding tissue. And once they invade that surrounding tissue, that healthy tissue is no longer going to be able to do its job correctly. When these cancer cells spread, it's called metastasis. If you've ever heard like commercials on the radio or on the TV and they talk about metastatic breast cancer, it means that these particular cells are their breast cancer cells. They're cells that are specific to the breast, but they've moved somewhere else in the body. So maybe they've metastasized into the stomach. So now when a oncologist goes into the stomach and they find these breast tissue, these breast tumors, then it's an indicator that, okay, this originated in the breast and has moved. So again, cancer cells, the reason that they're bad is they are preventing the surrounding healthy tissue of doing their job and absorbing the nutrients. So then they're taking all the nutrients, the surrounding healthy tissue is not getting the nutrients they need, and that will cause the cells to die off. Once those cells start to die, because they do have regulators on them, they can't just replace over and over and over again. So that will cause organ failure, and then that is what causes subsequently what could lead to death. So what causes cancer? It's typically there's some sort of defect within the gene of the cell. So it is some sort of mutation. Now, whether that is a mutation passed on from genetics, whether it's like down a family line, there are some cases of cancer that, like skin cancer, for example, if you have a family history of it, you are more likely to get it. However, there are things within the environment or things that you can control as habits that also increase your likelihood of getting cancer. So, some examples of that is smoking tobacco. Tobacco has been found to be a known carcinogen, which means a thing that causes cancer. Radiation is another known carcinogen. So, whether that's like too many x rays, or it could just be going outside and sun radiation, sun exposure. Defective genes, so again, getting that genetics from mom or dad or whoever, and then viral infection can also cause cancer. One of the common ones there is human papillomavirus, HPV. There's a vaccination for it because HPV is more likely to lead to cervical cancer in females, and it can also lead to, I believe, testicular cancer in males. So they want to make sure that gets taken care of. That viral infection is more likely to cause genetic defects within the cells and can then cause cancer. A damaged or defective what's called p53 gene. This is common in cancer cells, and again, it makes it so that it starts ignoring those signals. And so the growth signals are still trying to say like, hey, we need to stop, we can't keep going. But the defective p53 gene basically makes it so that it does not speak the same language as the growth signal anymore. So the growth signal is saying stop, but the cell is no longer able to respond to that. And so they just divide uncontrollably. Some treatments for cancer. Sometimes when cancer is found and it's found early enough before it has metastasized, those tumors can just be replaced with surgery. They can go in, they remove the tumor and some of the surrounding healthy tissue around it to make sure they've gotten it all. Then they will pull different samples of those tissues called a biopsy. They will pull those tissues and they will double check to make sure that there are no longer cancer cells present in that tissue. You can also use something called targeted radiation. This is radiation therapy. This is where there's a lot of math involved with radiation therapy, but essentially they look at the wavelengths of the radiation. They look at how deep the tumor or the cancer is inside of the body. And then they set up so that the radiation wavelength will hit that tumor perfectly, causing those cells to die and the healthiest tissue underneath of it to be okay. Chemotherapy is the third treatment for cancer, and this is going to be using chemicals or medications that have been known to stop mitosis in different steps of mitosis. So some of chemotherapy drugs, they can actually stop S phase from happening. So then that DNA no longer replicates, and those checkpoints that are checking to make sure that everything within the cell is fine, they say, hey, that DNA is not replicating correctly, like let's get rid of it. Now, because chemo does target cells that are going through mitosis, this is why cells that go through mitosis quickly naturally, so things like your skin, your hair, and your fingernails, this is why those particular cells will start to die. And then as a result, it is very common for individuals undergoing chemotherapy, depending on the drug type and how long, it's common for them to lose their hair and for their skin to kind of get a translucent discoloration. And that's because those cells that are responsible for the hair growth and they're responsible for the skin color, they are dying because they go through mitosis quickly. And the chemo doesn't know how to say, okay, I'm only going after the cancer cells. It goes after any cell undergoing mitosis. All right, so that is the end of Ed Puzzle 3. As always, if you guys have any questions, please let me know. Otherwise, I will see you in class when we can go over these materials together.